Okay, so let's now start with protein engineering. And if we talk about protein engineering, let's review the steps we need from a new catalyst to an application. So first of all, we have to find the new catalyst. How would we do that? So can you just give me a few examples? If we have an application, we have no catalyst in our fridge, we cannot buy anyone, how could we get a new catalyst? Do you have any suggestion for that? Yeah?
For example, metagenomics.
Metagenomics, this is like very intuitive, right? Easier.
Whole cells.
Whole cells, screening collections, right. So this is how we start. We can also take it from plants, whatever. And then we have to optimize it. And optimization means optimization of the reaction conditions, but also optimization of the catalyst. So, and then we should not forget this later. We have to formulate it. And the formulation means we have to stabilize it and prepare it in a way that chemists or the people who use it may use it. And chemists like to get a flask of white little spheres. They can put something and they can put this flask on the shelf and use it in a half year again. So this is where Luc comes into the game. But we should not forget that what we do here should be possible to be formulated and stabilized. And this is as important as the optimization. And this is more done by process engineers, and this is done by biologists, chemists, and biochemists. So here we have a very interdisciplinary communication. Finally, we have the biotransformation where we make a substrate to a product. And this is now where we get big. So actually we're talking here about one step out of 4, which is one link in the chain. If we want to have an enzyme for a new specific application, we need an active enzyme. We've heard of examples of enantioselective enzymes. We should not forget we have a whole bunch of properties that also need to be optimized. So for instance, we need, a high selectivity, and this is usually the reason why we use an enzyme. For instance, to cleave an ester, the easiest way is to use sodium hydroxide, but an enzyme might be enantioselective. So this is something we might want to improve, but at the same time, we need a high reaction rate because the reaction rate determines how fast the reaction is, or more important, how much catalyst we need. So, so if we increase the selectivity and destroy activity, we don't have a useful catalyst. Yeah? Catalyst should have a high area in this diagram. The next thing is the catalyst should be stable. So this means if we, for instance, reduce the activity like 2-fold and stabilize it 100-fold, we win. Okay, we're talking here about a multi-parameter set. Then we should have a good expression. If we optimize our catalyst and then we cannot fold it anymore, then we have a big problem. So it should be useful for the next step, the formulation. Then, of course, storage. Then something here which was addressed a bit by transaminases. This is not something which is not so obvious, but enzymes have been optimized by nature for very low substrate concentrations. Because most intermediates in the metabolism have a very low substrate concentrations. Some enzymes are different, but mostly evolution has optimized them for a low substrate concentration, and we want to work with 100 millimolar or higher. And some enzymes, actually for instance with transaminases, the substrate and product inhibition has prevented for years the application. Then we have here something, sometimes we need special properties. We want to couple it to a cascade, which is relevant for us. So maybe we need to suppress a side reaction, or we have to change the compatibility. And then we can do new things. Some of these properties you see here are extrinsic properties. Stability, for instance, can be influenced by putting the enzyme on a carrier. And for this, we don't need enzyme engineering. So if we want to stabilize our enzyme or increase activity, we have several possibilities to do that, one of which is protein engineering. Intrinsic properties like enantioselectivity depend to some extent from the reaction conditions, but mostly from the catalyst, from the amino acid composition. So some of these properties we can engineer with protein engineering, with other methods are cheaper. So for instance, if I want to stabilize my enzyme, I would not use enzyme engineering. The first thing I would try is to mobilize the enzyme, because this can be done within 2 weeks and not within half a year. On the other hand, novelty, like teaching the enzyme new tricks, like an inverted selectivity, for instance, or a new mechanism, Or for instance, we have a minor side reaction and we want to make it a major side reaction. For this, we do need enzyme engineering. So this is just a very practical example from an ongoing project. So this was just an enzyme reaction. I will tell you about this enzyme later. And we did several things. We wanted to increase the total turnover number, which means the amount of moles we produce per catalyst. So the expression optimization yielded an improvement from 100 milligram per liter to 160 milligram per liter protein. So this is roughly a twofold increase of activity. Yeah, the important point is, if you look here, which cultivation volume we need to get a certain conversion. So actually we should not use milligram protein in the total turnover number, but cultivation volume, because this is decisive how much enzyme we can make. So, and if we just make a conservative improvement of expression, or we go into another expression host which is more efficient, this can be much more efficient than improvement of the enzyme. Then, by enzyme engineering, we achieved a 6-fold activity improvement. This is very nice, because now we need 6 times less catalyst. But our collaboration partner, he immobilized the enzyme. He only got 20% immobilization yield, which is, I think, a fairly normal rate for an enzyme immobilization if you use cell-free extract, not purified enzyme. But he could use it 10-fold at last. So this is actually a 2-fold increase of the total turnover number. And you see from this, it is difficult to see which is the most promising. Because I would say this is a quite conservative approach, meaning we have so much technology available, it's so mature that we can expect an improvement. Here, often, we don't know. This also can be improved quite easily. So this would be the low-hanging fruits, and this is the risky one. Okay, so we should not forget enzyme engineering is just one method with others, and it's efficient once we talk about intrinsic properties. So, again, we can focus on reaction engineering. If you want to have a new catalyst, we can look for new enzymes. We talk about this, functional screening or metagenomics. This is a very fast-moving field. And I will talk today about rational design, which means getting an idea and changing an enzyme, or directed evolution. You will hear a lot about this in the summer school, so I will just present a few concepts, but you will get lectures by Uwe Bornscheu and others in a half year, focusing very on single aspects like high-throughput screening, mutagenesis, so you will get a very deep introduction soon. I want to make 2 points. So for an enzyme, function and structure is important. This is what is important for evolution, because the function is the important one and it depends from the structure. But there may be many sequences that encode for the same type of structure. There are enzyme families where every alpha helix is on the same place and you have 25% sequence conservation. So this means the important parameters are structure and function, and the one we can engineer is sequence. So this is a bit difficult. This is our screw. This is a point where we can change something, but this is not the actually relevant one. Of course, I mean, the structure is encoded by the sequence, but as I said, one structure might be encoded by many sequences. Then, if you talk about science, I think we assume that for every phenomenon, there would be one true explanation. So in science, you want to find out the truth. In engineering, we want to achieve a goal. We want to make a transaminase to accept bulky ketones. And there might be many ways to achieve this. We can achieve this with an error-prone PCR and getting many mutations, or we might find a magical residue in the active site with molecular modeling and change it. The result might be similar. So we do not care so much about the way we work, and we have to be aware we have several ways how we can achieve our objective. Because often we don't fully understand enzymes. So I would like to introduce you to the story of an enzyme, to have the full story of it from the initial idea to now recent experiments. And I just want to present you the story to see what methods did the people have available 25 years ago, what methods do we have now, what would be faster. And you're always in protein engineering in a certain situation. You have a crystal structure, or no, you have a good high-throughput screening, or no, you have a good expression system. And now we can see in which situations the scientists were at these steps and what decisions they made. So we start with the initial idea, then the screening, then how the enzyme was isolated, and yeah, they got a new catalyst. Then they dealt with the mechanism without having the crystal structure. And I want to show you we can do a lot of experiments if we don't have structural information, but this was really the breakthrough. Then they got the mechanism, Then they could engineer, and now we're talking about things like a commercial application, side reactions, and even the QMMM simulations were published last year. So the initial idea was, the objective was to produce profenes. Profenes are optically pure drugs. They are produced in the scale of several thousand tons. Interesting targets for enzymes. And there was an organic chemist in Japan who thought this would be a reaction to produce profenes from malonylates. So this is known in every organic chemistry textbook. You have an aryl malonic acid. It's not very stable. And an acidic pH, one carboxylic group is cleaved, and you get the racemate, because it can be either this one or that one. And he thought, many of the books I found in my organic textbook, we found in nature. So why not that one? So he thought maybe there's an enzyme that does this. He had no clear hypothesis about any aryl malonic acids present in nature. And then he thought many enzymes are enantioselective. So maybe this one will be enantioselective. So you have two big question marks. Does this enzyme exist in nature? And second question mark, is it enantioselective? If both are yes, then we have a new catalyst, which is highly attractive because we have an asymmetric synthesis. So he asked the student, please come here, I have this idea. Start to look for an enzyme that does the job. And the idea was, if a microorganism has this enzyme, it will be able to use the aryl malonic acid as carbon source. It can eat it and live. So they prepared minimal medium, and they grew the cells. And what they just did, they had a strain collection. And the PhD student, he went for the fridge, opened it, took out a few strains, made like microbiology work, single streak isolation, shake flasks. And then he looked for each single strain of all the several thousand strains if it grows, and they did not. So then he went back. This was the first year, and he went back for the supervisor and said, none of our hundreds of strains does the job. And then the supervisor said, no problem, go outside, go to nature, there's earth, take more microorganisms. And after 1.5 years more, he found a strain that could grow. And they put in a prochiral malonate, and they got out an optically enriched aryl propionate. And if it's optically enriched, usually it's catalytic. A spontaneous decarboxylation, of course, would give the racemate. So this was kind of evidence that there's an enzyme that does the job. So they were quite enthusiastic after 2.5 years. So just think now for a PhD thesis, how would you feel if you the first 2.5 years with absolutely no results? And, but they got it, so you see also sometimes it's worse to go for a great challenge instead of a conservative one, because you see he also got his reward. So what do we have now? We have a cell that does a conversion. We have no idea of the 6,000 or 4,000 genes, which one is responsible for this. So if this were your thesis, what would you do next? What would you do to find this catalyst? First question, maybe easier. How would you identify the species? What would you do to identify the species of a bacterium?
DNA isolieren und Blast machen.
DNA isolieren und Blast machen. Von der gesamten DNA? So I mean, like this is maybe 4,000 genes, maybe 6,000. Okay, guys, is there anyone with a biology background? How do you identify the species of a bacterium? You order primers, make a PCR, and make a 16S sequence the 16S RNA. This was now. In those days, they just looked how the cells were looking like, and they maybe they asked a microbiologist, and they found it was a gram-negative bacterium. Okay. Then, somewhere in the cell is a protein which makes the catalysis. And then also, there should be a gene. What would be your approach now if you were in the situation to get this gene?
Random integration.
What?
Random integration of
Yeah.
construct.
This is good.
When you cannot have colony, you made it,
Yeah.
you made it twice. Selected medium and
Yeah.
non-selected medium, and then you find that colony that goes only on non-selected.
Yes, this is an excellent experiment. What we do is we connect somehow something which destroys our gene with a way to find out if it still exists. So she suggested to use selective medium for growth. He was lucky. He had an HPLC method. This was the year when HPLC was introduced. He could determine the product. So he looked for product formation. But that's the same thing. So you should be aware, if you do this kind of research or want to engineer an enzyme or do something about it, you need to be able to have a good and practical method to determine activity. You should have something reliable. A high-throughput assay, for instance, like growth, can depend on many things. Product formation by HPLC detection is something reliable. He had this. And then we connect it to something where we knock out genes, and then we can see, okay, if it doesn't do the job anymore, we can we isolate this one. In this case, this would have meant a lot of HPLC results. What they did was, the inverted approach, they made the genomic library. You can also do this. You take the genome, make a genomic library, and then you look for cells who do the job. Okay, this would be a DNA-based approach. The one that he actually chose was a protein-based approach. He said on the protein level, he said there are 6,000 proteins. One of this is my catalyst. And now we need a good activity assay. So he purified the enzyme. And you see, oops, sorry for that. There was actually a table which explained the methods of protein purification, but only on my Mac. So he combined, of course, native enzymes don't have a his tag. So he combined 5 different columns, ammonium sulfate precipitation, anion exchange, and with the combination of the 5, and if you combine 5 columns, you don't do this in a week. He had to find this out empirically. So this is another year, and then he got a small band, and now he knew the first thing. The thing is, this is catalytically active, and we have 240, it has 29 kilodalton, so he knew the size. It's a very small protein. So now he started to get a bit knowledge about this. So if you have a protein in the SDS page, how can we get the sequence? Edman degradation. Great. This is what they did. So you need a lot of protein, and then you degrade it, and then you analyze the amino acids. And then he got the first 8 amino acids, and he could design a degenerated nucleotide. And with this one, he could look in his genomic library. This is how he found the gene. Nowadays, what would we do nowadays? I mean, Edman degradation is difficult.
You maybe go for MSNS?
Mass. Yeah, very easy. So 25 years ago, a lot of work. Now you just cut it out, go over the corridor, and give them this. Next day, you get an email with the sequence. So then he could purify the enzyme now. He could produce an E. coli. This is very nice. This is the most important thing for protein engineering. We need to be able to produce our enzyme in an expression organism. And then he characterized it. So he knew only the size and the kind of reaction. And then he got KCAT, KM. KCAT is nice. 300 units, 5,000 units. Pretty good. KM, he got in the millimolar range. What do you think, this is a high KM or a low KM compared to most enzymes, relatively spoken? Like, let's say in KM of 80 millimolar. What do you say? Maybe you can vote on this. So who's for high? 1, 2, 3, 4, 5, 6, 7, 8, 9. Who's for low? So good democracy. If you have a KM of 80, you need for the Vmax a concentration of at last 180 millimolar. This is terrible. In a cell, they will never have a 180 millimolar concentration of phenylmalonic acid. So, and this is something very, so he knew something. The substrate has the enzyme has a problem to bind the substrate properly. This is something not untypical or quite often observed for non-natural substrates. Don't forget, we have no idea why this bacterium had this enzyme. And this is for sure not the natural substrate. But we know the substrate binding, the affinity is not very high. Okay, so he went on with knowledge, but the initial idea was to use this to make proteins. He produced the precursor of a protein, made the conversion, and he got out an optical pure product. Fantastic. The problem is the biologic activity lies in the S enantiomer. This enzyme makes R. So actually, we talked about 2 question marks. Does nature have such an enzyme? And is it enantioselective? But the third question was, does it make the right enantiomer? So actually, after the PhD thesis, it was all for nothing. But he knew it's highly selective, and he thought, okay, maybe we can change this to make the right enantiomers. And this is something where we need enzyme engineering. It would be very difficult to use anything of reaction optimization to teach an enzyme to make the other. But so the first question is, which carboxylic acid group leaves? Now, think again. You're in the situation. You have this enzyme. You have no structure. You just know that it does a reaction. Do you know a way how to find out which of these two carbon dioxide groups is cleaved and which stays?
Radioactive labeling?
Radioactive labeling is strong, but we can also use carbon-13 labeling, and then we can use NMR spectroscopy. So, and this he did, and because he was Japanese, he did both enantiomers. And each of these two compounds is like a 5-step synthesis, so a lot of work. Maybe this whole thing was another year work. And then here we have the 13 carbon, and we can detect this by NMR. So what he found out is, if he uses this substrate, he has it still in the structure. If he uses this substrate, it's cleaved. So this was the evidence this one is cleaved. He had no idea why, but he knew that cleavage of carbon dioxide is not selective. So this means if you don't have a structure, you're a scientist, you're creative, you can design experiments and learn important things. Because once they got the structure, this was a very important information, which one goes, which one not. Because if you look now on the active side, you know this has to be destabilized and this one has to be bound somehow. Okay? So be creative. Of course, the standard thing is to get the structure, but biochemical experiments can tell you a lot. Okay, and then, wait, we're here in 1992. Nothing happened until 2008. Then they got the first crystal structures, so 4 or 5 groups simultaneously published structures. There were 2 or 3 empty enzymes. They didn't tell much, but there was also two structures with a ligand bound. And before this, there was absolutely nothing could be done about enzyme engineering. So this means the structure is a really important point if you want to engineer enzymes. Here's the active site. You see this is a carboxyl group that stays. That goes, and this is the one that stays. The one that stays has many hydrogen bonds in the active site, and the one which is cleaved is in the hydrophobic pocket. So they assumed this one is a very strong binding force. It lures the substrate in the active site, and then we have a very unfavorable interaction here. And this would explain the mechanism. Okay. Here we have a cysteine, and this cysteine can protonate from one side. And protonation from one side explains why it's enantioselective. So the rate determining step is not decisive for enantioselectivity. What happens is we get a planar intermediate, and this is then protonate from one side. So can we actually protonate from the other side? We can delete the cysteine and put one on the other side. So, and this is what I would call rational protein design. Because we have an idea, we have a hypothesis, we assume if this is the hypothesis is the protonation is the enantioselective step. If we can put the proton donor on the other side, then we would switch enantioselectivity. So we make a prediction, and we can confirm this with experiments. This is what simply is called rational protein design. And so this is a wild type. We have the cysteine 188. This was substituted by a serine, and they put the cysteine on the other side. And yes, they got the S enantiomer. So the prediction proved to be true. Okay, so if you want to make these kind of predictions, you need to know your enzyme very well, and you have to do lots of experiments. But then, if you know your system well, you can make good predictions. And now we have much more tools. We have like blast searches. We heard about this in the talks. We have bioinformatics. But somehow we have to be able to make hypotheses, and this depends on our understanding. This variant had a 10,000-fold reduced activity. So for some reason, they destroyed the enzyme, which meant they could predict selectivity, but they were not able to predict activity. And at this point, they switched the method. They said, okay, somehow this hydrophobic pocket is responsible for the destabilization of a carboxyl group. So let's modify it. And what they did was they mutated every residue, but they had no big idea which amino acid would improve that. We have here like 6 target residues, and 6 by 20, this is a lot of mutagenesis that has to be done. And I think you're familiar with the concept. Right. So they went for direct evolution. The first thing was they developed a high-throughput assay. In this case, it's easy. We have a prokaryomalonate with 2 carboxylic groups. We cleave carbon dioxide, so we get a monoacid. So we have a pH shift to the basic medium. And with a pH indicator, we can monitor the reaction. This looks conceptually very, very, very nice. And the assay pH assays, experience says this, are very unspecific. So they had to measure every plate 3 times. They didn't have much fun actually with this one. But they had a more or less valuable way to determine or to characterize libraries. So if we don't have a good idea, we need a good screening. If we have no idea and no screening, we have no chance. Okay, so what they did was, one after the other, they put all 19 other amino acids in each position. So they started with the serine 188, and they got a glycine, a 5-fold improvement. And then they used this as starting for, as template for the next round. And you see here, with different residues, they got improvements. And the best one was this one, and then another round here. So in 3 rounds, they got an almost 1,000-fold improvement of this enzyme. So you can see how powerful directed evolution is. And we change here a methionine into a leucine. In the pocket, this is a ligand. This is a very tiny difference. We change a valine to an isoleucine. And now we know there are other AMDases that are isoleucine here, but then we didn't know that. He changed a leucine to a methionine. They're very tiny differences. Why is this important for activity? We still don't understand it, but we are able to improve something we don't understand. So this is the great power of direct evolution. Here now at the actual state, this is how engineering is. So this is an S-selective variant, and this converts flobiprofen now faster than the wild type. So now we've achieved our objective. We recovered the activity. Also, you see these are several variants, and this is the dark one is naproxen, and the gray one is flobiprofen. And we see here that every variant is substrate specific. So this variant, for instance, the wild type, is faster with naproxen. This variant is faster with flobiprofen, and the improvement is only for the bright gray one. So the next thing was they used flobiprofen for this experiment as model compound, and they improved the enzyme with flobiprofen, but not the darker one with naproxen. So you see also, you only get what you search for. If they would have used naproxen, they would have found maybe mutants which have a higher activity with naproxen. Okay? So now this is last year. There was a group that published quantum mechanical calculations on the mechanism. So they defined a set of atoms in the active site, and they used a semi-empirical algorithm. And outside, they used molecular dynamics, which you learned this week from Florian. And yes, they found evidence for this indiolate. So now we know it's very likely that we actually do have, this was hypothesized, but now we have a very good hypothesis for the mechanism. So here you see the energy diagram, and you see they got a minimum here, this point for this planar intermediate, and this means this is the protonation step, the enantioselective step. You see the enantioselective step is indeed not rate determining, the decarboxylation is. And at this point, can somebody explain me the difference between a transition state and an intermediate?
Intermediate is something that you can, in principle, isolate.
Exactly. That's a local minimum. This follows the rules of chemistry. So in theory, even if it's very short-lived, we should be able to isolate it. They also investigated the possibility that it's concerted, which meaning leaving of the carboxyl group is simultaneous with protonation, and they didn't find a viable transition state for that. And because the calculations don't say it, we can assume that this exists, and this explains why we need here an aromatic residue. So now we have a pretty good knowledge of the enzyme. And now it can be commercialized, and actually several companies, including BASF, are investigating now these enzymes for commercial applications. Yeah, with the improved. So you really see the effect of protein engineering. 25 years after the discovery, we have S and R selective variants that can be used for commercial synthesis. So you really see that protein engineering makes a difference. Another thing is, what do you think? If we have the cysteine here, we get the R enantiomer. If we have it here, we get the S enantiomer. What happens if we have both cysteines? What's your guess? Like maybe the chemist, what do you think?
Yes, I have to think about it.
Anyone else? So here we get the S enantiomer. Here, this makes the R enantiomer. What happens if we have both?
Well, if they both the same active, I think you get a mixture.
A mixture, yeah, of course. And they got the mixture, and it was completely racemic. But you just said a very important thing, if they have the same activity. This is 10,000 times more active than this, and you get a racemate. And then they just put in the substrate, and they found out they made the racemates. So they interconverted one enantiomer with the other one. They don't need the melanin anymore. They can put an S enantiomer and get the R and vice versa. So they created a new enzyme. So you see with protein engineering, we can make new catalytic activities. There are racemases in nature, for instance, for amino acids. But there is no aryl propionate racemase. This is a really new enzyme. So, okay, and then they looked into nature. Nature has enzymes that have 2 thiols for racemization, but there are amino acid racemases. We can racemize only, like, for instance, glutamate. Aliphatic substrates, these are aromatic. So you see now this is actually something interesting to learn more about enzymatic racemization. And then one question, for instance, what is doing the reaction? Here we need a thiol that takes a proton, and the pKa of this one is around 12. In solution, cysteine has a pKa of 8, but in the active site you can model this with servers, and it was calculated around 12. So 8 orders of magnitude less. So what happens is, this one is activated by base, and here, sorry, this is activated, and this one gets the proton, so it can donate the proton. So we have co-catalytic residues. If you think back of the lipase mechanism, a similar principle. You see, in enzyme engineering, we have only 20 amino acids, but by the collaboration of amino acids, by the interaction, we can get new properties. We can get, for instance, a highly basic cysteine. Remains the question, what about this one? Here we have no co-catalytic residues. And another thing is here we have aliphatic substrates, here aromatic ones. And to find this out, the pH spectrum was studied. You see here. The wild-type decarboxylase has a very broad pH spectrum because the thiol is a proton donor. The glutamate racemase has a broad spectrum because we have this co-catalytic residues. This one has a very sharp pH spectrum. And at this point, this is something a scientist would say, wow, there's something. Because an explanation is it's exactly at the pKa of a thiolate. So this would mean the active amino acid is a thiolate residue. So this can be modeled by molecular dynamic simulations, and this is where Yasara comes in. So with Yasara, you can actually find out, is this stable? If you have a thiolate here, or if you have 2 thiols, what is more stable? And you see in the system with 2 thiols, one of them turns away, it's not stable. So this is where you can use Yasara. With what kind of enzyme did you use Yasara this week?
We only worked with one of the sample proteins because we had some problems. We didn't check before, but Yasara only runs with all the proteins if you have internet access, and we didn't have here.
Oh, really?
But we didn't realize before because we always run Yasara with internet before.
Okay. Anyway, did you do dynamics?
So I prepared some simulation because we didn't have time to run actual simulation, and we actually looked for the egg days. Okay.
So this is kind of, previously the semi-rational approach is very difficult. I would leave this to experts to collaborate with them. But MD simulations is something you as experimentators can do. And from your basic lectures, you have the knowledge. You can actually read how these programs work in Wikipedia. Everything is written. It's a very, very easy physics, but you can really learn something. And now, once you have a model, you can try out, for instance, the effect of amino acid substitutions and develop hypotheses. This one is, we thought we need deprotonation, and once a substrate is bound, there's absolutely no water. This we can learn from molecular dynamic simulations. The empty enzyme is accessible. And this is one, this is now semi-empirical calculations. It's an actual reaction path. So now we can see and get the full mechanism, and this is a kind of good evidence that we need a thiolate. So if you want to do rational design, get to know your enzyme well and make a lot of experiments, then you will be able to improve it. So just one slide of it. By NMR binding experiments, residues were identified, and again, saturation mutagenesis improved a 40-fold activity increase. Okay? Without going too deep, is if you make rational protein design, find a method to get ideas and develop hypotheses. You do not need to make accurate predictions. Here, they just identified relevant residues, and then they saturated them, and they could also improve the enzyme. Okay, we just go over this now. So if we look back 25 years, there was a completely new enzyme discovered because a professor had an exciting idea. Maybe this professor had also other ideas we never read about, but we read about this idea. And they found the gene and could produce an E. coli. This is now possible for most enzymes, so it's a luxury for us, but this is the most important thing if you want to engineer your enzyme. The structure determination was a milestone. So if you want to engineer an enzyme, first thing, look at the structure information and download the paper and read it. Yeah? Not all crystal structures are the same. Some have good information, others have in some parts of the enzyme very poor information. Okay? This is what's determining your success. And if you use engineering, we can invert selectivity. We can teach the enzyme to make a new activity. We can now explain the enzymatic mechanism. And we can increase activity. So far, we still don't really understand why amino acid exchanges influence, or how they do this, how they influence activity. The second thing is, we still have no idea about the natural role. So this was just one example how we can see which are important points. And if you work with an enzyme class which has been heavily engineered by others, then you can rely on this. So there's some groups that work 20 years with the same enzyme, and they can make the most exciting tricks with it. But the big challenge is, if you have an interesting catalyst, a new enzyme, maybe a new enzyme class, and you're the very first one to engineer, then you are in a situation like this PhD student where you have very little information. And then you should know how you can get to know your enzyme quickly, and with which methods to understand it, or how you use randomized methods for improvement. Okay? So they're very different challenges. If you improve a transaminase, there are many papers about transaminase improvement, but maybe not about your fold. So others, if you improve a lipase, there are thousands of papers about lipase improvement. So always your situation is very different, and the literature basis and your database is very different. So this is a very frequent challenge that we have first to see what is the available information. So the basic problem of enzyme engineering now is we have a primary structure. So what do you think? How well can we predict a structure from a mere primary structure, a sequence? What do you think?
I think in the primary structure, so we should look how many cysteines, so the molecule, how many cysteine residues, so there are how many cysteines, so there are in the molecule, in the protein sequence, and then from this, so we can predict the secondary structure.
Yes, to some extent. There is a very fairly accurate secondary structure prediction. And I would say until 15 residues, 20 residues, small peptides, we can excellently predict. Maybe you refer to also statistical analysis. So usually, for instance, residues have a helix propensity. We would rarely find a glycine in an alpha helix, but we would find, for instance, a certain motif in the alpha helix. Yeah?
What do you want to do with the secondary structure?
Excuse me, the three-dimensional structure. Yeah, the secondary structure is the first thing. So if you have a small secondary structure element, you can predict that one. But you're right, an enzyme is composed, let's say, for instance, of 20 helixes. And if you can predict everyone with 80%, it's not so high. But sometimes it's useful to be able to predict certain elements. Okay? I just want to give to your attention two approaches. One thing is what we take a small peptide and we perform like a dynamic simulation, an all-atom approach, and we simulate the folding. The second thing is we compare the sequence to databases. We have a PDB database that's amazing. And then we can see, for instance, methionine, valine, methionine, valine is like 99% in alpha helices. So always, if we want to understand, we can use comparisons, like blast searches, or we can use simulations. But as a matter of fact, for small peptides, simulations are amazingly good. So a zinc finger can be very well predicted. But as soon as we talk about an enzyme of 200, 300 amino acids, it fails because we have too many degrees of freedom. Yeah, I just want to call to your attention, there are many people who work on de novo predictions and they're not bad. What would be the pragmatic approach? If we have an amino acid sequence of a new enzyme, how can we get an idea of the structure?
Just blast it and then see if something that is similar has the structure. And
Yeah.
then you can do some modeling on that.
Yeah, we just compare it to other structures. How reliable is this?
Depends on the similarity or identity.
What is a good similarity?
Yeah, I wouldn't go below 60.
No. With other enzymes, like I have an enzyme in my laboratory, it has 25% identity, and every helix with other enzymes in the same place, if you crystallize it. Depends on the enzyme. Yeah? But the point is, if you don't have an experimentally determined crystal structure, we have a big problem. It's very difficult to predict here, and a homology model is not a prediction but a comparison, so it's a very rough. It will help you, for instance, to identify of a transaminase the residue which binds the PLP, most likely. But for instance, if in the AIM days problem, when we have this tiny difference between valine and methionine, a homology model is worthless. It's useless. Once we have the structure, can we predict activity? In a certain sense, yes. For instance, a lipase, then we can say, yeah, it's a lipase. We have a catalytic triad. But I see you shaking your heads because it's very difficult to know which substrate is converted. For instance, we find an enzyme, this is an esterase, and we say, yeah, it's an acetylcholine esterase from a mammal. What about butyrylcholine? If we make the substrate a bit bigger, can it be converted or not? The only way to find this out is trying this out. So if you want to engineer something, we want to make predictions, and we have two weak points. First of all, it's very difficult to predict structures. And once we have the structure, it's still difficult to predict function. So the rational approach is still very challenging because we don't understand enzymes very well. So this is just about the diversity. I mean, we could just try out all variants, and this is a very easy calculation. If we have a peptide of 10 amino acids, and we put in every position all 19 others, so we only make single mutants, we have to screen 190. If we want to make double mutants, we have to screen 16,000, 190 by 190. So you see, even with a trivial problem, and I would say like the best variant of AIM-DAYS is a 6-fold variant. So if you want to check all possible combinations, you have no chance. Now this is a more normal enzyme. So here we get 3,800 variants if you want to check all single mutants. So what do you think in a master thesis? How many mutants you can characterize? If you have to plan a project, your PhD student your supervisor asks you, how you get this master student, how many mutants you can characterize. What would you say? What's realistic? Pardon?
I know. I think that would be possible in 2 years.
This in 2 years.
Maybe a bit longer.
How would you approach this? Would you do this by PCR?
No, I know a PhD student who had such a protein, and when he was finished, he had all those.
Yeah. What you can do is, for instance, you can order a library and synthesize the genes. But still, this is a high-throughput thing. Okay? So rationally, this is almost impossible. And if you want to screen this, you can order for, I think, for 10, 15,000 euros a library. And if you have a good high-throughput screen, you can check this. Okay? But in my opinion, this is possible. This is already direct evolution. You will not be able to purify each single variant and to make a thorough characterization. No? This would be more a high-throughput approach. But nevertheless, think of AIM-DAYS. Most single variants are useless. The best ones are just double variant, and then we are out of it. So a systematic characterization of the diversity we have is very difficult. So this is how people like to show it. It's called sequence space. So all the possible genotypes, they encode for a so-called fitness landscape. So this would be the different variants that are good with one properties and have other properties. And we have to navigate in this sequence space. We want to go from one highly active variant to the next one. And these are hikers, so you see the speed, if you do this by hand, with whom you're walking through this. They have an advantage, they can see peaks, we can't. So this is a point we have to navigate in a huge diversity, and as soon as we talk about double or triple variants, the diversity is totally out of scope for any rational-based approach. So these are the mostly shown approaches for enzyme design. I told you this one. Here we get an idea. We make a site-directed mutagenesis, and you characterize. We just saw an example. One idea, one mutant, success. Right? This is the ideal case. The problem is here. We don't understand the enzymes. This is with us the problem. Then the next thing is we randomize it. For instance, we order a library where you have all 19 other amino acids in each position. And if you want to categorize 3,800 variants, you need a good high-throughput screening. And this can be experimentally very easy or very challenging. How do you want to make your mutant screening? If you work as transaminase, how would you do that? Can you give me advice?
I think it depends. Do they have a few screens available, like the alpha MBA?
How many variants you could screen in a week with an alpha MBA test?
I don't know.
What do you think?
I mean, it's a multi-wall screen, so probably not that many.
So if you can make 5 plates a day, you have 500 to 2,500 per week, right? But 2,500 per week does not mean 50,000 per week. So some screens like this one, is maybe like a medium throughput screening, would be efficient if you narrow it a bit down. if you make a pure randomization, you should screen several 10,000s. But a lot of assays, for instance, are only good for several hundreds. And this means we have to narrow down our diversity by using rational elements. So this is a problem. Most assays, like the pH assay, are not fit for a real high throughput screening. With a good robot, you can screen 10,000s. You will see a good robot in Uwe Bonschar's lab in Greifswald in September. And then an industry is doing this, but this is also very costly and usually out of the scope, for instance, of a master thesis. But like 5,000 colonies, you can screen by hand. This is quite easy, okay? But for 5,000 colonies, you need to narrow it down. So usually, we have not so good high throughput screenings, and we have a not so good understanding. So we have to work somehow in the middle of the both. So, and this is conceptually, if we have no understanding, we use random mutagenesis, and we have a huge screening effort. If we have a very good information intensity, we have a good knowledge, we just put the cysteine on the other side, we make one mutant, and we get it. So we have like a screening effort of 1. But in most cases, we don't have a good crystal structure. It's a new enzyme. Then it's interesting. So we're here. We have some knowledge. Maybe we have a homology model and can identify the active site. And then we need a medium throughput screen. This is a normal case, and this is called focused directed evolution. This is done mostly, and this is quite efficient. You saw this with the AMDASE example where they just mutated the active site residues. Okay? But with each enzyme, you first have to identify where you are. Do I have a lot of information or not so much information? If you have no information at all and no high throughput screening, the best thing is start with the crystallization. So here is a picture that we have. It summarizes different methods that are around. And I told you now about error-prone PCR, for instance, randomization. In this case, you do not need any understanding, and you get a lot of many mutations distributed over the whole enzyme. Here, you get single variants. There are also something where you shuffle genes. What I want to show is all of these methods can achieve the objective. But you have a different result. Here you have many mutations, and all of them would have very likely to make a small contribution. Here you have very, very few mutations with a big impact. Okay? So don't forget, the method, the way you choose, determines your result. Okay, this is actually the approach I'm talking about. It's not so new. 1984, somebody says, if we have a mutant that can produce a variant that can produce a mutant spectrum, and it's self-reproducing, meaning phenotype and genotype is coupled, and that's what we have in a cell. In a cell, we have the protein and the DNA, and they are kept together by the cells. So once we have the enzyme, we can find the DNA. Phenotype and genotype are coupled. And then we can amplify the clones, test the clones, test for optimal phenotypes, and then go back to the genotype and either choose this one or make another round. Was in 1984. So what we're doing here, or what some of us are doing, is 30 years old. Right? So these are a few companies, just to give you an overview. So for instance, Codexis, I think, is present to everyone. They, for instance, everyone is not talking about the Codexis transaminases, and they were engineered. So this means people are really making money with improving enzymes. Actually, what we're doing, this is a potato farming. This is how potatoes looked when Columbus came. This is how they're looking now. And what happened was that the farmers, whenever they took the harvest, the next year they would take the nicest and roundest for the next round. So actually, if you're strict, we're doing a screening. What we do is more like plant breeding than like true evolution. Yeah, because evolution is a very, like we have many actually fitness parameters for survival. And here we're just looking on one. Okay? So, if we make it very simple, we have something which is non-sufficient. We have certain variants, then we make a variation, and then we get a repertoire, and we have to restrict it. Restriction can be we make one new mutant, and we characterize it by gas chromatography, or we make 5 millions and characterize and make a selection by FACS or with a growth assay. But this is, if you boil it down, what is protein engineering. You have variants, you have variation, rationally based, mutation, or randomized, and you have a restriction. Then, since now every problem requires its own solution, because in most enzymes, the information density differs. Then, the method determines the outcome. So, at the beginning, when you choose your experiment, you should be aware that it has a very strong impact about the result that you will get. Then something, fancy method, fancy name. For some reason, everyone in this field is putting names like sesame to PCR methods. And so if you hear the name of the method, you have no idea what it is. Yeah? So what you should do is if you develop your method, think of a fancy name which has nothing to do with the method. And finally, this is very important. I told you when the decarboxylase was improved with flubiprofen, the best mutant was better with flubiprofen, but not with naproxen. Right? You get what you screen for. Don't forget that. Okay. So the first time I want to start, or maybe we can just make a 5-minute break now, and afterwards we can, for the last half hour, go over through like technical approaches. Okay? Thanks. Okay. Shall we go on? Are we complete? Yeah. I saw nobody leave, so we just go on. Yeah.
About you get what you screen for.
Mm-hmm.
What if you have your substrate is not easy to in order to have an easy assay to
Yeah.
screen for the enzyme, if your substrate is not suitable for that,
Yeah.
can you choose a similar substrate?
Yeah. It's called surrogate.
For example,
Yeah. Let me just go for one slide, then I can answer your question. So we want to ask if you want to screen for a substrate which is not available, right? And then you design something which is similar. This is a good idea, right? Yeah. So this is an example from literature. And this is exactly what you were asking. This is one of the very first works of directed evolution. And this was antibiotic derivatives. And they had to cleave this ester for some reason, industrially. And chemically, this is not easy because you would destroy the beta-lactam ring. So an esterase would be cool, it works nicely, but Harald told you about a high space-time yield. And for this you need to dissolve your substrate, and this one is not soluble in water. So they had to add co-solvent, and the co-solvent inactivated the enzyme. So in the early 90s they said, yeah, this is a great challenge to prove our principle of protein engineering. So this reaction, you could make now improve the esterase for activity towards DMSO. If you cleave here, you change the pH. But I just told you, a pH assay is not something you would really like to do because it's very unspecific. So this one is a bit difficult to detect. And then they thought, okay, this looks very similar. But if we release this one, we get paranitrophenol, and the paranitrophenolate anion is strongly yellow. You will work with this in Greifswald. This is a very easy screen and the standard reaction for lipases. So this is exactly this case what you said. This one was synthetically available, but difficult to detect. So they said, we have a surrogate compound. And they got a 24-fold increased variant with the nitrophenyl ester, which was only 4-fold increased with that one. So what is the difference between these two compounds chemically? Pardon? Yeah, between like chemically, between this compound and this compound.
The oxygen and the hydroxy is not para, isn't
It's it's on a methylene group, right? Yeah, this is an alcohol, this is a phenol. Looks similar, but of course, this is an extremely good leaving group. Good leaving group. This is an alcohol. This is a pretty bad leaving group. So this is an activated ester, and this is why this compound, this ester, is something totally different for the enzyme than this one. It's just that the structures look very similar. So this was why actually the enzyme was improved for the good leaving group, but not for the not so good leaving group, right? So, but to answer your question now, this is a compromise you have to do. So in this situation, you could say, we do this one, but then we have a poor assay. For instance, like pH. And then we can screen, we have to screen every plate 3 times, so we can maybe characterize 500 variants. This one is very, very, very sensitive, so we can use the robot and we can screen 10,000 variants per week. So you can maybe in your whole master thesis or PhD thesis in 1 year screen 50,000 variants, but you go the risk that you cannot transfer it. So we're talking here in protein engineering about experimental constraints and theoretic constraints. And my advice is you should have a good analytics available to characterize the true substrate. This is the most important point. So the first thing you should do is get an HPLC method, a GC method, GCMS, where you really can show, okay, I can get units and KCAT, KM of my system. And then you can use a surrogate substrate, and maybe this will produce false positives. So what? then you just go on screening. But you should be able to confirm what you get. So if you read a paper, check, did they publish only high-throughput screening results, or did they publish true product information, for instance, measured by GC/HPLC? So if you have to work with a surrogate substrate, you can save a lot of time. But for instance, then you should check your mutants and confirm it with the true substrate. Okay? So, but I like this question because this is what the direction we're going to. In protein engineering, we are talking about practical and experimental constraints. Okay, I go back. So, I just want to just discuss now quickly a few concepts we need for protein engineering. The first one is how to bring in the mutants into the genes. So, we can do this chemically with mutator strains, or also what's called error-prone PCR. And this was for some time the mostly used one. Any of you ever has worked with error-prone PCR? Me neither. Because it's a lot in the literature, but it's quite difficult to do. With some genes it works nicely. In some cases we add manganese to this. The transformation works often poorly. This is why you suggested to use a different cloning method. So this is a widely used method. What we do is we make a PCR, and we do some tricks to reduce the fidelity of the polymerase. We use a polymerase without proofreading. We add some other metals to disturb the enzyme. And with this one, we just get a lot of mistakes. So far, so nice. Great advantages is freedom to operate. There's no patent on this, so if we get a nice advantage of this, we can earn money with that. So this is something companies like. The problem is ligation. So we once tried to establish this with for lab course. We got 1,000 clones for the transformation for the normal PCR, control reaction, with the error-prone PCR 3. And then we thought that this was too unstable for lab course, and we discontinued that. So the ligation, for instance, for some reason is very difficult. And I will talk a bit about non-statistical nucleotide exchange now. So, you know there are two kinds of mutations. We can mutate in base to a similar base or to a different base. And if we talk about polymerases, we're not talking about letters like GATC. Yeah? To understand this, we use letters, but these are chemical molecules. This change is quite easy for a polymerase to happen. This doesn't really happen. And the polymerases do this differently. So you always have to see transitions, same to same, happens often, to transversions. And you see there's only like one transition possible from A, but two transversions, right? So this would mean a polymerase is likely to exchange A by G, but not by T or C. And this is called bias. So mutagenesis, we're not talking about informatics here. Mutagenesis is biased because we're talking about chemical reactions. So this is a very difficult to understand slide. This is a systematic approach of comparison of different polymerases and the mutation rate. So if we have all exchanges in the same likelihood, we would have 8.3 for all of them, right? So these are the possible ones. In the ideal case, here under this real conditions, A to G and T to C happens each with 30%. So if you have a library, 2/3 will have only transitions, and some are very underrepresented, some even do not happen. So the Taq polymerase is nice because it's cheap, is heavily biased. Of the possible biochemical mutations, you will get only very few. These are very underrepresented. But this one is interesting. This is a Fu polymerase without proofreading. And here, they got a very equal distribution. So this means if you make an error-prone PCR, you should be very careful about the selection of your polymerase. Because here we get a lot of mutations. Here we will get always the same mutations again and again. And if you screen 50,000 variants, you want to know, do you screen 50,000 different mutations? Or do you screen 50,000 variants and 30,000 are the same? Right? So this is called the biochemical bias. And it's really worth to try out different polymerases and to read about this and check this out before you start screening 50,000 variants. The next thing is the genetic code is degenerated. You know, for instance, alanine, 4 different codons encode for alanine. So if our polymerase changes this G for A, we get the same. So actually, in an error-prone PCR, you will produce quite a significant part of wild type. Statistically, 20% of your mutants is wild type. So of the 50,000 variants you screen, like 5 weeks, 1 week you are spending screening wild type. Right? So this is actually, if we have this codon, let's say we have we introduce 5 mutants per gene per round. You can adjust this with the conditions. It will be extremely unlikely to have 2 mutants in the same codon. Yeah, if you have 300, 900 nucleotides, it would be extremely unlikely that you have something where 2 adjacent codons would be mutated. So we can neglect this. And with single codon exchanges, from alanine, what do we get? Valine, glutamate, glycine, proline, serine, threonine, and a lot of wild type. Nothing else. Nothing of this. So error-prone PCR is not a true randomization. You get only a very small part of the cake. This is actually a real mutagenesis of GFP. These are all the possible mutations, and they sequence the results. These are all they got. So you see, for instance, about isoleucine, they got like half of the possible. So statistically, like you can access from each amino acid only 6 of the 19. Okay? So this is a very limited method, but on the other hand, if you have no idea about your enzyme, this is the best method to use. But you should be aware of, there's a lot of literature about codon bias, and if you should do this, it's very worth to spend like an afternoon reading about this. It's better than 6 weeks screening a library which is full of wild type. Okay, the next thing is mutation frequency. I want to know, like, I want to have, this is an important parameter. If you're too heavy, too many mutations per gene, you kill the enzyme. If you're like too few, it's very difficult to get multiple mutations. So this is something you should try out. But what we want to have it uniform. This is a realistic distribution. So this is what we want, 1. But if we have 1 per gene, 30%, these have too many, and this is wild type. So you see, we don't get a sharp distribution of 2 per gene, but we would get like a distribution. And so what you can do here is, for instance, nowadays, you can just order a library in a company. And the company can actually adjust in the synthesis, and they can introduce mistakes. They can make this more uniform, but this is really expensive. So this would be maybe 10,000 euros. And you can think of maybe you, so in our project, we have like maybe 20,000 for everything cost per month, per year. And you can think of spending half for a library or doing this in one PCR experiment yourself, right? But so we can adjust this, but buying libraries is still something expensive. So another approach is the quick change PCR. Who of you has done a quick change PCR? 1, 2, 3, yeah. So this is, I think, fairly easy. And what you can do, so I will not go into the details. What you do is you use primers with bearing mutations, and you amplify a whole plasmid. Then you get a kind of linear, 2 linear fragments that are circular, make a circular form. And this is transformed. This is nicked DNA, so the transformation efficiency is really poor. When you made your last quick change, Raquel, how many clones did you get?
I don't know, but a lot of them.
A lot. Positive?
Yeah.
You?
It doesn't matter. I mean, I think I had a few, but not that many. But you just need 1 clone.
Yes, in a quick change. But now we can use a primer where we put in N and N. N means all 4, right? And so N means all 4, like one of the all 4. So in the synthesis of the primer, they usually they put, yeah?
Can you explain that to some biochemists?
Yeah. So, so you know that GTC, for instance, stands for codon, right? Like GATC. G-guanine, so. And N means all 4. So when you order a primer for molecular biology, the company who produces it, they make a cyclization. So they add nucleotide to nucleotide, G, A to C, right? And these are consecutive reactions, work up, reaction, work up, reaction, work up. Like in a chem lab, only there's a machine who does this. But then, if you have N, the company will add G, A, T, C in equal proportions. So you will get 1 primer with a G, 1 with an A, 1 with a T, and 1 with a C. And this means, statistically, if you have this, and you make this 3 times, you have a wobble codon. And this wobble codon encodes now for all amino acids. Or you have all possible codons in this. You would have, for instance, one molecule will have GAG. The other molecule will have CCC, right? And every one you put 25% of each. And this we refer as N. So N stands for this that they put in the mixture of all four. So if you buy a primer with N and N, you buy one with all 64 codons encoded. So, and if you do this, then we get, of course, a cyclization product which has in the position of the X all possible codons. So we can, this is how we did this with the AIM days. We mutated all amino acids with such a primer. And then it makes a big difference in your transformation if you have 3 or 3,000. Because you can calculate library size. For instance, here we have 64 codons, so we need at last 64 variants, right? But actually we have some we will have double, others we're missing. So we say rule of thumb is possible number of variations by 3. So with this one we should screen 180 variants. So after the transformation we should get out 180 variants. If you have 3 variants, 3 clones, you should make this many times. And I just tell this to you, quick change in many cases works very smoothly, but in some cases you have a poor transformation efficiency and you might lose some weeks to optimize this. And this can be different from gene to gene. So this is a molecular biology challenge you have to solve. And you can Google this, there are a lot of tricks. For instance, don't transform BA21, transform DH5 alpha. Yeah, so easy, it works perfect. So, but sometimes this needs attention. But and then usually you are able, like usually these libraries are between 100 and 5,000, and this is a very efficient method to produce it. Okay? So in this case, for instance, this variant was done with a quick change, and now, okay. So the choice of codons is, this is a bit you can even. How to say this? You can restrict it even more. So N stands for G, A, T, C. So K stands for, let me see here. I think it's, what are we missing here? TG, right? K stands for T and G. So here you see all 64 possible codons, and I told you it's degenerated. So if we use the K, which only encodes for 2, we cleave half of this, and this means we only encode for half of them, right? But we still have all 20 amino acids. Yeah, we have the yellow codons we still have. For instance, we have here C, G, T. This is how we read it, right? We have still arginine. But we don't have this codon for arginine. But it means we still have all amino acids represented, but with half of the codons. The formula to calculate coverage is this is a rule of thumb. 95% coverage means my library contains with 95% statistical probability all amino acids. Okay? And this means number of variations multiplied by 3. This is how you get this statistical likelihood. So in this case, we have 64. So to make sure we have all 20 amino acids in here, we have to calculate 64 by 3, 180. Here we have 32. So in case of K, so here we have N equals 64, N, and K, N, and K equals 32. So 32 by 3 is 100. So we only have to screen half. So the difference between 100 and 200 colonies is not so dramatic. But what happens if we want to mutate 3 amino acids at the same time, right? So then we have here, case A is 64 by 64 by 64 by 3 compared to 32 by 32 by 32 by 3, right? So think of this. This means we have the same number of amino acids present in the library, but we have to screen many more here, right? So Just the point is, we're not going too deep. If you make a saturation mutagenesis or very single residues, sit down, check out this, and you can use appropriate codons. So for instance, NNK is only 20. NNS, for instance, has a different set of nucleotides. And also, you can make even stronger restrictions. You can, for instance, only take, in case of K, only like D3 of them or so. And then you can make a set for only hydrophobic residues. And now we're talking about biochemistry. If you have an alanine, you can make a set that only encodes for 5 hydrophobic amino acids. You say, I don't want to have an aspartate. You make like an aspartate, you make it like in a blast search of your transaminase, and you say all transaminases have only leucines, isoleucines, and valines. So we really don't need an arginine there, because it will most likely be inactive. And then you can say we only put 3 in there, right? 3 amino acids. So we have maybe 8 codons. So you can reduce the library size, and this means reduce the number of codons you screen. So why do I say this? So if you randomize, you have to make sure what the bias, how you introduce your mutations experimentally, how they're distributed. If you make site-directed mutagenesis with variation, you have to look a bit into the math, and then you have to decide how big can the library be, and how much I want to focus it, how many residues I want to change. So on the one hand, you have to think, how good is my high-throughput screening? How many variants can I screen? Can I screen 5,000 or 50,000? If you can screen 50,000, you can make much bigger libraries. You can be much more creative. But if you can only screen 5,000, then you should restrict this using degenerated codons. And this is how you have to design your experiment. And this is a very decisive phase of your experiment, because this will decide what you will do the next 6 weeks and if you will have a hit or not. Okay, so in this case, for instance, I showed you of this amazing activity increase. They put in 20 in each position. All hits they had are hydrophobic. They mutated a hydrophobic pocket and they got only hydrophobic residues. So we can, for instance, here, isoleucine, methionine, phenylalanine, leucine, isoleucine. Yeah, of course, it's a hydrophobic pocket. There are no other residues tolerated. So if they would have been a bit smarter, they could have restricted this to 5 hydrophobic amino acids in each position and saved a lot of work. And then they can screen 2 positions at the same time. And this is the point. When you make your experiment, look at the result. If I were to continue this, I would only use very condensed libraries. No use to introduce arginine there. Of course, before they couldn't know that, right? So look at this, plan your experiments carefully. You can save a lot of time. Okay, this is just this coverage thing. So if we have like 1 amino acid, we just have to screen 100 variants to get 95% coverage. If we have the NNK codon and 2 amino acids, maybe 2,000 variants do the job. But 3 or 4, we have tremendous numbers. Yeah. What you can do is we can use NDT. I didn't go into this. NDT encodes for 12 amino acids. So we have only 12 codons. So you don't put all the 20 amino acids, you just use very restricted pools. And you see, you miss some amino acids, but 2 amino acids is 100 variants. So the question is, do I want to have a large variation in one position? Or do I want to vary several positions at the same time with restricted sets of amino acids? And to make this decision, you need to know the structure of your enzyme very well and to know what you're doing. Okay? So, the last thing is, experimentally, if you have a library, you made an error-prone PCR, you made a quick change mutagenesis, how can you know if you are successful? I mean, what happens if your experiment went wrong and you only have wild type? You don't want to spend half a year screening 50,000 times the wild type. So what will you do? You can sequence. But sequencing, maybe with Sanger, you can sequence maybe 10, 20 variants. You should do that, but this is still a lot of work. What you can do is you make 1 plate and you screen it. First use the wild type, and then you will get this distribution. So you order the clones according to their activity. These are the highest active, these are the lowest active. And then what you get here, this variation, of course, is the quality of your assay. Right? So this actually is a very poor assay. And then you take a second plate. So if you want to screen your 50,000 clones, first you just screen 1 plate. And then you put all the mutants according to the activity. This was a library using saturation mutagenesis. And then What happens here? Half is inactive. And that's good news, because we have mutants, and the mutants do something. The point where we put the mutants, this has some effect on the activity. It's negative, but we have something. If your mutant landscape looks like this, you're very likely to have either you have wild type or you're using the wrong mutagenesis method. Okay? And those here, those will be the candidates for an improvement. So this means if you want to screen a large number of clones, start with screening one plate and see if something changes. And if not, then you should sequence a lot and look for the problem. But this quality check is worth the work before you start screening like thousands of variants for nothing. And this is called the wild type landscape. The wild type landscape tells you how good your assay is. And the mutant landscape tells you if your mutagenesis was successful. Okay, the last thing I want to talk about is a few concepts about screening assays. Let's say we have now a nice library, and don't forget, the way of making the library decides how many clones we should screen. So we have different approaches, and this axis shows us the samples per day. So if you prepare in shake flasks, and if you're very hardworking, and you purify your enzyme, maybe you can characterize 10 variants per week. Really good. If you go for the microtiter plate, one plate is 96 variants with controls 90 variants. And I think if you're good, like in a centrifuge, for instance, you can put 4 plates. With microtiter plates, if you're really good, you can maybe screen 8 plates per day. So this would be 800, so maybe if you're really good, 2,000 per week. Then you can put an agar plate. For instance, you can very easily use a selection to determine if your cell has a kanamycin resistance. Right? And with these approaches, selection, you can screen, think of agar plates, 50,000 within a month. Very easy, right? Then you can use a fluorescence-assisted cell sorter. This is, for instance, if you use for your high-throughput screening something which makes fluorescence. Here you can characterize hundreds of thousand variants, even more. And then if you have growing cells, that's the most, you can just use fast-growing cells. So you see, different approaches have very different throughput. What my recommendation is, you should get a slow throughput method where you can exactly say we have product formation. This is very important, and then you should get a fast method. Okay, so one thing is, another concept is inactivity pre-screening. I just told you many of your variants will be simply inactive, so you don't need to screen them. You can make it kind of active. For instance, with the esterases, you can, this is a tributylene agar, and if you have an active colony, you get a patio around your colonies. And this means this is an active colony. If you have this, you have maybe 1 day more in your experiment, because you need to pick them and put them on this pre-selection plate. But if you have a library of 50,000, and you can remove the 15,000 inactive variants, you save a lot of time, right? So this might be worth the work. The next thing is, if you do this, you will identify those that are not correctly folded. So really, you make it a kind of filter, only to filter, so to get through the nice and interesting variants. Okay, so if we talk about high-throughput screening, it should be very sensitive. I just told you the pH assay, and if you have an assay like this, you will spend a lot of time with repetition. Then it should be selective, so you should avoid side reactions and enzyme cascades. It should be feasible with cell-free extract. Right? If you do it the next day, you should get the same result. With a high-throughput assay, you need a lot of manual skill, so check yourself. So you should be quantitative, so you should be able to put units at the end and not something like absorption. Okay, so you should know how many units you have in your wells. And you should cross-check it with other methods. So I repeat again, the first thing is you need a good epPCR method, and then you compare it to your assay. So the best thing to establish an assay is to purify enzyme and use purified enzyme solutions, you know the specific activity, in the microtiter plates. Sometimes there are surprising differences in the microtiter plate to units measured by epPCR. So the substrate. This is an important point. Is it available? We had this question before. If you need a 5-step synthesis to make your ideal screening substrate, then you will spend a lot of time making these 5 steps. So you could look for something similar that you can buy. Or you should know some collaboration partner who can do that for you. Then you will depend on him. I had a friend, he was working with a race group, and the race group produced a screening compound for him. until the epPCR broke. And then he had to wait half a year until he could get the compound again, because the only guys who had this preparative epPCR method with the chiral column to separate the enantiomers was the race group. So this is a very practical approach, but I see the success of high-throughput assays depends on practical aspects. Then the principle of measurement, and this is very important. You said you have several screening assays available for the transaminase. Keep it simple. You will get enough problems with the library, with the handling, so the measurement principle should be simple. If you have a complicated measurement principle, you multiply your problems. So, endpoint or kinetic? This is a question to you. What do you think is better? You have something. Harald said chemists like endpoint because they made a batch overnight, and biochemists like kinetics, right? In a high-throughput assay, you can do the same. You have your property, for instance, like your absorption, and you have your time. So what you can do is you just wait overnight and you get one measurement point. Or you can make a kinetic and you get the way there. What do you think is better?
Yeah, maybe if you want to screen for a really active variant, it's better to measure the kinetics, because if you wait overnight, and then most of some slow variants will also have the same conversion, because if you have a full conversion, then you can get more conversion, and you can screen for the really fast enzymes.
Yeah, it's even worse. We don't know how we came here. Maybe you have a problem with heating your plate, and it's like this, because the inner wells need some time to come for the reaction temperature. Or the variants have a low stability. What you have is like this, right? High-throughput screening curves sometimes look weird. So if you can measure kinetically, you should do it. Sometimes you have an assay by back titration, or you have to add a reagent. Then you have endpoint. If possible, use kinetic. The drawback is you will get pretty big tables in Excel files you have to evaluate. Okay? So last point, any expensive instruments required. So I read the paper a couple of years ago for a high-throughput assay with a flow-through NMR. And the author said he will expect that this is used in any laboratory in the near future. In the acknowledgement, he asked Bruker for lending him the flow-through NMR. Not even he himself had a flow-through NMR. Okay? This means for you, if you have a friend who has a fancy device, you make a high-throughput assay, all those who also have such a fancy device will cite you. If you make a new measurement principle which is very robust and you just need a microdata plate reader for it, companies will do it, many people will quote you. Okay, so in academia we should develop simple and practical assays. Okay, I will go through this. No, I have no time for this. The last factor is just a practical thing. Start with the wild type. Actually, don't start with the wild type, start with purified enzyme in your plate. If all the slopes look uniform, then use cell-free extract. Sometimes this is not possible. If cell-free extract works, then cultivate your cells in the microdata plates, and then you should get something like this. These are activities in the microdata plate. And your mutants should look like this, right? Sorry for the German, should be landscape. What you should do then is to check statistically if your assay is right. That's the last thing we talked today about. So this is, for instance, a positive control. This is a negative control. You determine the standard deviation, and you determine your signal. So this is this would be your standard deviation. This would be the difference of your negative control and your positive control. And without going into an example, if you have time, I recommend you to calculate mu p equals 6, mu n equals 1. Which means we have a signal of 6 with the active enzyme, 1 with the empty strain, for instance, and we have a standard deviation in both cases of plus minus 0.5. And then you can calculate if this is feasible. So if the Z factor is smaller 0.5, doesn't work. If it's bigger 0.5, then it's robust enough. That's the rule of thumb, easy to calculate, but this is another check you should do. And the last slide for today, you saw it already, is this one. And I just showed this to you because this is a very nice strategy. In the 1994, the first time they made direct evolution with pyronitrophenol. You can look on the reaction you do, and if you find any way that your enzyme reaction releases pyronitrophenol, you won. then you have an assay. So if you develop a high-throughput assay, you should try to bring it to a known measurement principle. And you see here the very important point, the assay is always prone to practical constraints. In this case, availability of the compound, or better, the possibility to detect the compound. So, to wrap it up, in enzyme engineering, on the one hand, we talk about understanding the enzyme. And you could see in the first part, if people understand the catalyst well, we can make hypotheses and get good predictions, good results. But in most cases, and those that are scientifically interesting, we have no crystal structure, and the enzyme is quite new. We have not so much experience. And then we can use randomized methods. In randomized methods, we have to make mutagenesis, and this is quite heavy molecular biology. So you need a couple of months to get to know these experiments. And then we need high-throughput screening, which is quite heavy on the analytics. So it's very nice. We work together with understanding of enzymes, with analytics, and molecular biology. You get a very round way, very round training. Okay, and with this, I'd like to open for questions, and thank you very much.